Okay. Yes, thanks and welcome everybody. I've managed to pick up a lurg, so I'm hoping the voice is going to make it through the presentation. So sorry about that. And today I wanted to talk about keeping coccidiosis under control with a real emphasis on preventing the disease. So, doesn't want to advance, we're doing well here. So what we're going to talk about today is understanding coccidia. So what classes of livestock we're going to see disease in, what symptoms might we see, how we can diagnose it, and what we can do to treat sick animals. And then looking at preventing coccidiosis on your farm, because I'd much rather it's seeing it prevented than be treating it. So what is coccidiosis? Coccidiosis is a parasitic disease caused by a protozoa, and the genus is called Eimeria. It's really common in young sheep, cattle, and goats, but clinical disease is generally associated with underlying stress. If an animal just gets exposed to a small number of coccidia, generally they will not get sick, develop an immunity, and we don't see this generally in older animals. But when we do get a big outbreak, it can lead to severe economic loss. So with any disease, the key to prevention is understanding it. So most of this talk is going to be on understanding coccidiosis, and then we'll talk about how we use that knowledge to prevent it. So as I said, it's caused by this protozoa called Eimeria, and there's lots of different species. The coccidia are host specific, so that means that the cattle coccidia are not the same as goat coccidia, not the same as sheep coccidia. And there's between 13 and 20 different species for each of the cattle, sheep, and goats, but they don't cross-interact. Under a heavy load, you might get a small amount of infection in a different species, but not enough to cause clinical disease. And because the animals develop an immunity over time, it's mainly younger animals where we see the issue. So the life cycle is quite important, and understanding that will help preventing it. And basically, the coccidia pass oocysts, which are eggs in the feces, and these contain 8 infective, what are called sporozoites. And the sporozoites become infected, so we call that sporulation, within 1 to 2 days, but it can be up to a week or 2, depending on the temperature. Once they're infected, they can be ingested by a susceptible animal because they're exposed to fecal contamination in their environment. And once in the abdomen, into the stomach, that oocyst is digested and releasing the sporozoite, which then infect the intestines. And they invade the cells and then they multiply. And they go through a whole series of invasion, multiplying, and then the new ones will reinfect other cells within the gut. So because of that, we can have really, if you get a high level of infection, you can have really significant damage to the GI tract. And that a lot of the time that damage can occur before any more oocysts are actually produced. It takes about 16 or 17 days from when the oocysts are ingested to when more oocysts are produced. But in that time, there's all this replication going on inside the gastrointestinal tract, which means that we can have some pretty sick animals, animals that are poor doers, but we don't actually see those oocysts in the feces for a few days. Once the oocysts pass in the feces, as I said, they can be infected as quickly as 1 to 2 days, and that's at 25 to 30 degrees centigrade. So the warmer, moister climates, we often see a bit more of a problem with it. Where you've got cool or very hot weather, it can take longer for those oocysts to develop. And freezing or prolonged exposure over 40 degrees centigrade can actually kill the oocysts completely, as can dry conditions and direct sunlight. So they are relatively fragile, but in the right environment, they can persist for a long time. So in those moist, cool conditions, it's particularly when they're shaded, they can survive for several months to over a year, which means that, in a cooler, so Tasmania and Victoria, southern Western Australia, where you can get some cooler shaded areas, then you can get reinfection from year to year within if you've got a seasonal calving or seasonal lambing, you can actually still have oocysts lasting from the previous year. Whereas in your drier, hotter environments, they will be killed within a few weeks to a couple of months. And where you've got really high temperatures and direct sunlight, they can be killed within a few days. So this means that within Australia, we see a little variation in how it's presented. So in your inland area where you've got high temperature and drying and desiccation and lower stocking rates, we generally don't see a problem with coccidiosis until it rains. We put animals in a yard and we get crowded, wet conditions. And then because they often haven't been exposed to it at all, those weaners can actually be quite susceptible to it because they haven't previously been exposed to a very high load because it's killed in their environment so quickly. Where we probably see a lot of problem is more your coastal areas. And I note that a lot of people here from New South Wales. I think this is definitely a problem we see a lot in New South Wales, where you've got warm, wet conditions, where those oocysts will sporulate within a couple of days and infect animals really quickly. So you can get a rapid buildup of the disease. But what we don't see is generally carryover from season to season. You'll always have a few adult animals or weaners or older animals that have low levels of coccidia, and these will introduce or maintain them in the environment, but they don't cause a rapid buildup unless they're all crowded in together. And then in your temperate southern climates, where you get more consistent rainfall and often more mud and cooler weather, then it survives for longer. So where you've got intensive farming and higher stocking densities or cold winters, we can see more of a problem with it. So we generally see symptoms, as I said, in younger animals under stress. So the most classic symptom is diarrhea, which is often bloody and is quite irritating. So you'll see them straining and swishing their tails. And so I'm not sure if you can see the mouse up there, but you can see there's like a butterfly effect showing up on the bottom of these lambs here. And that's a bit of a classic sign of it being coccidiosis rather than anything else, because they wiggle their tails around a lot. And so same with calves. And so you'll get quite a lot of feces staining either side of the rectum, and it forms like a butterfly shape. You'll often see weight loss. The animals won't grow very well because they've got all this damage inside their intestines. They can be pot-bellied because they've got fluid within their gut. They may be dehydrated. Severe cases can be weak. They will sometimes have reduced feed intake. And in really severe cases, they can die from it. However, it's not the only cause of diarrhea in young stock, which is why it's important to get a diagnosis and know what the problem is. Obviously, a major differential is worms, and often we see coccidia in conjunction with worms. And generally, once we've treated the worms, the coccidia goes away by itself because that's the major stressor. We'll often see it associated with Salmonella or Yersinia, which are also diseases we see when there is stress and high stocking rates. It could be another differential would be bracken toxicity, which causes a bloody scours. In calves, we see pestivirus causing scours. In lambs, we see campi. Generally, with the Campylobacter scours, they occur in a younger lamb, but you can also see coccidiosis in lambs of 4 to 8 weeks when there is severe stress. And another differential would be giardia. So important to actually know what you're dealing with and get a diagnosis. I'll talk more about how to do that later. So what are the predisposing factors for the disease? So really 2 things. One is a high environmental load. So anywhere where you've got animals in yards, around stock camps, then you've got a build-up of mud, which allows the who assist to survive much longer in the environment, leaky water troughs, and even in water itself. You can get infected feed, or when feed's fed out on the ground and then you get fecal contamination of your feed. When stock are kept in barns, etc., it's often very commonly found in bedding and generally where there's a high stocking rate. So when you get a flood, for instance, or it's a drought and you've got to get everything into a yard, then it's time to be thinking about, is there coccidiosis a risk if you're dealing with younger animals? And then the animals itself, anything that will reduce the immunity. So overcrowding and mixing of different groups of animals. For some of those lambs and kids that get it at a young age, if they haven't had, or even calves, if they haven't had sufficient colostrum, then that may still be having an impact. If you've got major mineral deficiencies, and again, any form of stress. So weaning, particularly early weaning. So if you've got lambs with a big age range, so you're weaning them at 12 weeks, but some of them are 4 or 5 weeks, those younger ones are going to be much more stressed, much more likely, because they're younger as well, to get coccidiosis. If they're transported, if you have floods, severe weather, they're cold, they're miserable, they're in a drought lot, they're malnourished, they've got worms or other diseases. Any of those things, if I see coccidiosis as a diagnosis, I always want to know what else is going on in that flock or herd, because generally the coccidiosis is a symptom of something else that is wrong. The other thing to remember is that the younger the animal, the less coccidia it will take to make them sick. So our younger animals, those that are weaned earlier, those that don't get colostrum or under nutritional stress, it takes less coccidia to get them quite sick than it would do older animals that are already beginning to develop an immunity. So one of the things you can do as a control is to actually not have a wide age range of young stock, because you'll often get older calves or older lambs that are shedding it. They're not sick, but they will be spreading it around, and then the younger animals pick it up. And because they don't have the same immunity, they get sick. So how does it present? Well, it presents slightly differently in our different animals. So really commonly we see coccidia in lambs post-weaning. And again, high stocking density. We've got a high worm burden, so really important to have an effective drench and know that your drenches are working. If they're in feedlots or any sort of form of intensive feeding regime, if there's stress from transport, any change of diet, particularly a significant change of diet, upsets the gut, makes the animals aren't digesting their food as well, they're more likely to get coccidia. Severe weather, where they're all getting together and bunching up in a muddy area, or you've got to put them all together, or they're just cold and they haven't got enough food. And anywhere there's a contamination environment, particularly by carrier ewes. So ewes and goats, but particularly goats, but also ewes, will shed coccidia around the time of kidding and lambing. Cows will also have a slight increase around the time of calving, but particularly this is something we see in goats. Once they've been infected, lambs normally have a solid immunity, so we don't see this as an issue in our older sheep, but they will keep shedding it for a while, even though they have got good immunity. In cattle, we often see this in dairy calves at that 4 to 8 week of age. So, as I said, it takes about 16 days before the coccidia have gone through the full cycle. So generally, we don't diagnose this less than 3 weeks of age. And it's often a disease we see in at 4 to 8 weeks of age. In calves, whereas in lambs, we see it a bit older because it's when they're weaned. In beef calves, we often see it at weaning as well, particularly the earlier weaned calves. And we can also see it in feedlots when they're under stress and mingling, etc., which is why generally feedlots will put something like monetsin into the feed to try and control that. Stresses are exactly the same as sheep, so transportation, high stocking rates, diet changes, concurrent disease and weather. We often see coccidia in goats, and one of that is like certainly the goats I deal with are smaller flocks. You've got a wide age range of kids, so you'll often have 4 or 5 month old kids as well as very young kids, and so those older kids will be shedding quite a lot of coccidia and infecting the young kids, and also you get periparturient rise. So the goat dams will actually be, the does will be actually shedding more coccidia around the time of kidding. So much more of a risk there as well. And so the young stock get infected, and then 5 to 8 weeks of age, by the time it's got its cycle, we'll see disease in young kids. Also in goats, we see less rotational grazing as a general rule. So you're more likely to have areas where you're going to have coccidia build up within the environment. So how do we diagnose it? Well, certainly you'll see the symptoms, but it's important to do a fecal float to confirm the diagnosis. This is exactly the same as you do a worm egg count. So any lab that does your worm egg counts will also be able to give you a coccidial count or tell you if there's coccidial there. If we've got severe cases, then we'll diagnose it on post-mortem. And you can repeat the fecal floats to monitor, but there's not a clear relationship between the count and the disease severity, because often, as I said, you can get severe disease before you even see the oocytes coming out in the feces. So there is, for each species of animals, there is an area where we have a cutoff, but it's quite a wide range because of that fact that not always the oocytes have come through, showing how severe the actual clinical disease is. Okay. So how do we prevent the disease? Well, what we want to do is basically think of all those risk factors and put in preventive measures to get rid of them. The most important thing, I think, is to actually preempt when you're going to get coccidiosis. So we all know that when you get a flood, but weaning, etc., if you know you've had it before, you know the symptoms you're going to get, to actually look at some of those preventive strategies before a problem arises. Our basic preventive strategies are to manage the environment, to reduce the stress. We can also use prophylactic measures and biosecurity practices. So environment, basically removing or minimizing all those sources where there's lots of feces and lots of mud. So having clean paddocks with low worm and coccidial burden for putting our weaners into, making sure as possible, as much as possible, we have dry and well-drained areas. And if we've got yard animals, it's dry and well-drained in there as well. Rotational grazing allows stops build up in those camps, etc., where we find that animals lie around a lot. So if we don't have camp areas, then we're less likely to get issue with coccidia. And as I said, it survives a lot longer in shade and mud. So to try and minimize those, not have leaking troughs, that sort of thing, will get rid of the problem. And, with calves, for instance, in dairy calves, it's often in the huts, etc., where it's shaded. So to move areas around where you've got calves, move your shelters and expose the underlying areas to sunlight and heat. Clean water, particularly in troughs. Obviously, you'll often get a build-up of coccidia around the edges of dams. So preventing any mud build-up, not having leaks. And when you're feeding out, and you're feeding out on the ground, then to move those sites around the paddock so you don't have this one area where you always feed out and there's lots of manure around that area where animals are also eating. With lambs, we want to reduce the stress at weaning. So having enough feed, having clean paddocks, Again, having that tighter lambing period, so you don't have those little baby lambs that bit younger and more likely to be more susceptible. To know that your drenches are working, so to do regular drench tests so that we don't have a worm buildup. And also because one of the ways we can control it is by feeding various ionophores, etc., in the feed, to have sheep that you can, if you need to, put onto a grain source, so using that imprint feeding pre-weaning, so that lambs are used to eating a grain if you think that coccidia may be something you need to control. So very briefly, talking about those prophylactic measures, there are anticoccidial feed additives or medications that you can use for high risk periods. So these would be common to include for dairy calves, for instance. But obviously, a lot of people don't feed grain to their weaners, but if you are, then it's definitely something you could include, particularly if you've got to lot feed, or if you have got a flood or something like that. So you can include monensin or salicin into the feed. The important thing to remember with this, though, is it needs to be used continuously for 2 to 3 weeks before it's effective. So this is one reason why you have to predict a problem and start feeding it before you have animals that need treating. We also have a product called Coccidiosis, and that is a single dose by mouth. Again, it needs to be given a week before the expected outbreak. It's only registered for cattle, and it has quite a long withhold period. So it's something that can be used, but you need to preempt the need for it and remember that withhold period. The different coccidia can also come onto your farm, so by buying stock in. So having good biosecurity practices so that you're not mingling animals at that important area or important time when particularly like you've got weaners, etc. So you're not buying other animals in or mixing animals from different properties at that time of stress. So having a quarantine process where all animals brought in and put in a yard, put in a special quarantine paddock, and that's not where your stock are going, because obviously they've got to develop an immunity. So if you introduce animals at a time of high risk, then it's more likely to cause a problem. And you also want to I've already said that, so avoid that high risk period. How do we treat it? Well, again, immediately we want to try and isolate those animals that we know are affected, because they're going to be producing a large number of oocysts and affecting other animals. And we always want to look for that underlying cause. So I would recommend that you would consult a vet for your proper diagnosis and a treatment plan. We may need to give really sick animals fluids, electrolytes, etc. And there are anticoccidial drugs, so the totrosuril can also be used as a treatment. And there are sulfonamides, which are antibiotics. These can be an injection, but can also be given in feed and water. Again, there is no product actually registered for sheep, so any vet using it would be using those oral products off-label and would need a prescription to do that, as you would anyhow, because sulfonamides are an antibiotic. So yeah, if you have a problem, then you really need to be talking to your vet and working out what the best treatment is available in your situation. If you want to know more about coccidia, the Parabos website has got a few resources, which Fiona's going to put in the chat, I believe, in a minute. So under cattle, you can search coccidiosis, but in sheep and goats, it's under protozoa. If you just search the Parabos website for each of those species, you'll find a bit more information coming up there. And to summarize, the most important thing for controlling coccidia is to be proactive about your prevention, know when you're likely to get a problem or you're at risk of getting a problem, and making sure that everything else you do has happy, healthy, productive livestock. And if you've got no stress and minimal exposure on your farm, you're unlikely to see an issue with coccidia, or it's unlikely to be a significant problem. Always understand. So the most critical thing is to remember that the oocysts can be infective as quickly as 1 to 2 days. They last in the environment for anything from a few weeks to up to a year. And once the animal has been infected, there's this cycle of multiplication and infection that goes on inside the gut, which means that our animals are quite sick and have a lot of gut damage before we even see symptoms. So managing your environment and reducing stress is critical. And if this is not possible, to actually think early and introduce those products to control your coccidia. Also to remember that your animals can be quite sick before it's possible to diagnose a problem. So if you have more questions, I know we're going to talk now, and there's some great resources available on the WormBoss site as well.
Great, thank you, Alison.
I think my voice just is hanging in there.
Your voice held out. Alison has done a few miles and hopped off a plane just this morning from overseas to present to us. So we're very grateful. She's running on, I'm sure, lack of sleep and has picked something up on the plane.
Oops,
So we now will dive into question time. And if you'd like to put your questions in the Q&A panel now for Alison, and we'll pop through the questions and finish off. So the first question that's come through, Alison, is in a contaminated paddock, where can you find the oocysts? Is it in the grass or the soil?
Anywhere where they've been, so unlike worms that crawl up the grass, as the larvae hatch, the oocysts are basically anywhere where there's fecal contamination. So it's going to be where there's mud or feces around the place, or where animals are eating close to feces. So they stay within anywhere that's muddy. So they obviously come out in the feces, but if the feces get spread out and trampled around into mud, they will remain in the mud, but they like that moist, wetter environment. They're not going to come crawling up the grass or anything.
And the next question we have follows on from that. So how does the oocysts die off compared to, say, a worm egg when you're trying to think about cleaning up your paddock?
They're probably not dissimilar, really, in that, I mean, like worm eggs as well. We know that if we can dry it or desiccate it or spread out and dry out the poo, then we can get rid of the worm eggs. The same with our oocysts, really. So it's probably not, like worm eggs can survive an awful long time in that cooler, moister climate, and so can coccidia. So they're probably pretty similar.
Next question that's come through is, if you suspect coccidiosis in a cohort of lambs or calves because there is an indication of scours, what percentage would you expect to be actually infected? Is it just a single animal in, say, 40 or more of the flock or herd?
Generally, if you've got it there, it'll be almost all of them. Like every single animal will, at some point in its life, is likely to get exposed to coccidiosis, okay, or coccidia. Most of them, if they're not stressed, will just have a small, they might be off color for a couple of days. We wouldn't even notice it. And then they keep going and they develop immunity. So when you've got animals with clinical signs, it's probably the tip of the iceberg. But if it's only the younger animals that have got signs, the older ones have probably already had it and they've got over it. But if it's the older ones that got signs, then the younger ones are most likely to get it, and you could end up with more problems. So it's like any disease, including the lurgy I picked up on the plane on the way home. You get exposed to it, you develop immunity, you hopefully don't get it again. But anyone that's sick, there's always going to be other animals there that are definitely exposed but don't actually get sick with it.
Yeah, thanks, Alison. Next question. Many studies reported for coccidiosis in goats from Australia. Do you know?
Sorry, say that again.
Do you know of many studies about coccidiosis in goats that have been done in Australia?
I'm not sure about in Australia per se. There's definitely studies that, there's papers published on coccidiosis in goats. But I'm not sure about in Australia, but there's definitely coccidiosis in goats in Australia. So, yeah, I'm just not sure of any particular Australian studies I can think off the top of my head. I'm sure they're there.
Just a couple more questions here. How can we correlate the oocyst count and clinical coccidiosis?
So there's some guidelines out there as to the levels that may be indicative of clinical disease, and that varies from species to species. But because there can be significant clinical disease before the oocysts actually shed, then an oocyst count may not be predictive. You could have much worse disease there than you realise, I guess. But for any particular animal, there is a cutoff at which point this is indicative of clinical disease, and you put that count together with what you're seeing in the animals, I guess.
And last question. Oh no, here we have a couple more coming through. Is there any way to take rumen fluids from immune animals and drenching young susceptible animals to help develop their immunity? It's an interesting question.
Interesting question, and not that I'm aware of. And I'm not sure. Because the immunity is actually within the intestinal cell wall rather than the rumen fluid per se, I would say, I think. So because they invade the actual villi, so within the intestine you have these finger-like projections which are highly absorptive, and they invade the villi of the intestines, and so the body, rather than the inside of the guts, is providing that immunity once they've been exposed. So potentially there could be a vaccine developed, maybe, but because we don't see it as an ongoing problem once they've been exposed once, I don't know. And because there's lots of different types of coccidia, I don't know, how easy it would be to develop a multi-coccidial species vaccine, or whether there's even the call for it, considering that generally you can control coccidia by getting everything else right.
Thanks, Alison. That was a nice curly question, but good, well thought out there from the person who asked it.
Yeah, I just think that the important thing to remember about coccidia, it's an indication there's something else wrong with your system. So the important thing is not be focusing on trying to treat your coccidia, but going, why have I got coccidia? What is going on with my system that is causing my animals to get coccidia in the first place?
Is there genetic differences in the host, and can there be selection in a high challenged environment?
Correct, there is, and that's probably something else I could have said. There is definitely, yes. So one of the things I did read somewhere when I was doing some reading for this talk was, it's suggesting that the animals that don't get sick are the ones you keep. Again, in most situations, I think you can manage it without having to go to selection, but certainly those animals that don't get sick are likely to have a better immunity to coccidia, or they could just be older. So that because you've got that age interaction, the older the animal, the likely it's already had a low level exposure, so the better immune system is. It's a bit harder to pick that. But if you had a cohort of animals all at the same age with minimal previous exposure, then the ones that don't get sick are more likely to have a better immunity. But in reality, on a farm,
thanks,
all at the
Alison.
same
And I
age.
know you have a it's the question about organic systems. And have you had any experience or anecdotal evidence of using oregano extracts or other products in organic systems?
No,
And
I was
thank
trying.
you for that
I
question.
had that there as a in case this question came up, and obviously someone saw it. I was trying to mute myself so I could cough, but that didn't work. So no, I have no experience with it. I found it on the internet. There is a published paper. I think I referenced it. Hang on, let me see if I can find it again. I think I referenced the published paper. Yeah. And I can probably follow up on where that came from and have a look at the reference. I haven't even looked at it. The other thing is that, tannins are generally meant to help as well. But the important thing to remember about tannins is that they also binds minerals, so you can get mineral deficiencies if you do use tannins. So no, I haven't, but there is a paper published on it, so I can find out a bit more about that reference if you want me to.
Thank you. And the next one is hold on, just checking I have not already asked that one. That might have been a bit of oh yeah. Pathogenicity is different for each Emeria species in goats. So is there any method to differentiate morphologically between these species? And this question was asked in two different parts, so I hope I've put it together correctly for the person.
Yeah. Yes, you can tell. Generally, if we actually go back way back, you can tell underneath the microscope what the different species are if you're good at it. So this paper, I took that illustration from, just went past it. These are actually the different Eimeria in goats. And as you can see, they all look slightly different. So you can, underneath the microscope, get a reasonable idea, like they're different sizes, some of them, okay? Some of them have got these little cap areas here, and some of them are completely spherical. So there is different pathogenicity between the different Eimeria, that is correct. And you can get an idea of which is which underneath the microscope. I suspect there's probably also PCRs for it as well. But again, it's not something we, I think because they're easy to see underneath the microscope, that's the way we generally do it.
The final question I have is they would like to know more about nervous coccidiosis, but haven't mentioned anything in particular there. So
yeah, again, that was the other slide I hid because I wasn't sure it was very relevant, but I thought I might get a question on it. So there it is. Radio. So basically, in the odd occasion when you do get an outbreak of coccidiosis, you might, and not always, get an animal that has neurological signs. And those are uncoordinated movements. They're generally down, when I've seen them being down with their head back in what we call ophistotinus. They can have tremors, convulsions, seizures. They may head press, and you can have blindness. We're not really sure exactly why you get nervous coccidiosis, but it's thought that when you get the severe intestinal damage, there's release of toxins or maybe some immune-mediated compounds that are released by the body to help combat the coccidia, and these affect the central nervous system. And so in these animals, you'll get the scarring, but you'll also get the nervous systems as well. So it's not, as I said, a common presentation, but it is something we do occasionally see associated with coccidia. And generally those animals die, they're pretty hard to treat.
Great. Thank you, Alison. And a special thank you to everyone in our audience today for asking so many varied questions. That was a very interesting question time. That brings us to the end of today's presentation. So thank you so much, Alison, for jumping off a plane and being tired, exhausted, and not very well, but still presenting to us a great presentation on coccidiosis.
Sorry about the voice. It really wasn't doing me any favors, but we got there, so all good.
Excellent. Thanks everyone for joining us, and I look forward to you joining us at next month's Paraboss webinar. Until then, have a great week. Thanks very much. Bye for now.
Bye, everybody.